Hey guys, American Infidel here, and today we're gonna do something a little bit different. I know you'd much rather watch me go and probably shoot a gun and do a review on it, but today we're gonna talk about something called DNA. Now, I just know talking about it will get pretty boring and you'll probably leave the video immediately, so let's try and make it interesting by visualizing it. So, I'm gonna pull up GTA, and we're gonna get on this. Okay, so here we are on GTA 5, and we're gonna do some science. Okay, so, first of all, I think cars are the best representation of DNA, and let me show you why. Yeah, there we go. Get out of the car. Oh, there goes. No, we don't need that anyway. Don't worry about that. We got our science suit on, we got our science boots on, and we are ready to science. Now, if you look, DNA is a set of what we call codons, which are made from 3 bases to create an amino acid in sequences. These codons include many letters like a code. Yeah. And these letters are A for adenine, T for thymine, G for guanine, and C for cytosine? That word. These letters are called bases, or nitrogenous bases. And the car's tires are like the bases, you see? Look at that. The bases. 1, 2, 3, 4. 4 bases. Oh, I probably shouldn't have that in the video. The tires are like the 4 bases. 1, 2, 3, 4. 4 bases. Correct? Right. Now all we're missing is, well, how the hell are those DNA? Well, if you look here, DNA, right? Attached. So imagine those blue stripes are the parts that attach them, right? Okay, 1 second. Yep. Oh, right. These bases have to go by the base pair rule, which states A can only pair with T and G can only pair with C. Now imagine the tires of the car are the bases, and they are linked by phosphate groups or phosphate and 5-carbon sugar. When a base is connected to sugar and phosphate, it's called a nucleotide. Now these nucleotides connect in long sequences to make DNA, deoxyribonucleic acid. These long sequences, these sequences connect in long strands and twist into a double helix. Kinda like traffic. You have no idea how long it is. One strand is about 2 miles long if stretched out and is called a chromosome. Now, if you took all the DNA from your cells, it would be as long as the diameter of the solar system times 2. I would not like to be in that traffic. Now DNA makes RNA, ribonucleic acid, by splitting in half. So let's try and split the car in half, shall we? Now let's get there. Now we wait. Here it comes. Now remember, this is for science. We're gonna split this car to make RNA. 3, 2, 1. Impact. That didn't do what I wanted it to. One second. We can make this work. We can do this! Damn. Guess we'll just have to wait for the next train. We will split this car. Trial 2, Day 1. Car 1. 3, 2, 1, impact. Where's my train? I need my train! Train! We will do this. Don't worry. Everything is not lost. We just gotta be patient, is all. There'll be a train someday. Right? A train. Trial 3, Day 1. Car 1. Impact. In 3, 2, 1. Impact. This is not what I wanted. I wanted destruction. Well, I gotta say, I'm starting to think DNA isn't like a car. Unless a car turns into 2 motorcycles. Of course, I shall turn this car into 2 motorcycles, and it will work. Oh wait, no, RNA is single-stranded. All I need is 1 motorcycle. I see. So it is settled. I shall make this car a motorcycle. Well, I guess this roundabout over here shall do. If I don't get it stuck. Are you ready? Ta-da! I did it! I've turned the car into a motorcycle. I've turned DNA into RNA. Okay, so we made RNA. Not quite. RNA is made by splitting DNA and pairing them again, except instead of an A to T, A binds... No, not A. Yes, yeah, A binds to U instead of T. So U replaces T. U meaning uracil. So let's say we have a sequence going ACTA. That RNA, the RNA of that would be UGAU. But even then it doesn't work. Why? Don't tell me I forgot about start and stop codons. Start codons are codons that determine when a sequence of DNA is started, and a stop codon is when it stops. Everything between is junk, and we don't care about that. Wait, I forgot about amino acids. Okay. Okay, amino acids are made up of a sequence of sulfate groups made up from the 4 bases between the start and the stop codon. Example, a start codon is AUG, then an amino acid is GUA, then a stop codon is UAA. So the sequence is UAA. The sequence is AUG, GUA, UAA. The amino acid in the middle is valine. Okay, back to RNA again. DNA makes RNA, makes protein, makes bacon. This is the central dogma of stuff, and it makes up your genetic code and genes. Now when RNA makes protein, it helps build muscles! But first, how does RNA make protein? Well, RNA goes into cells, you know, with the cytoplasm and everything. Anyway, these cells read the RNA and make the corresponding protein. And that, my friends, is the central dogma of science. DNA makes RNA makes protein. But wait, what's the GTA equivalent of a cell? What does a card do? Go inside of, split, be read, and then make this. And then how is this read to make something? Wait a second. Explosions! Flip over. 1 second. Yep. No. Maybe. Damn it. Okay. I'm gonna try this. We will get this. Don't worry, guys. I got you. Don't worry about it. Okay? Okay? Let me show you why. Great. We are not going to let off the gas. That's what we're doing. Oh boy, we're letting off the gas. Nailed it. Yeah. Missed it by that much. That was sudden. Nailed it. This is not the right place. Oh, there it is. Don't mind me, everyone. Just driving in a completely wrong direction from where I was supposed to be. Well, guess that's happened more than once. Apparently I'm up against this corner right now. There's like a full 5 inches there. Shut up, Bruce. When the physics break in GTA. I'm not even touching this board. Now we just keep going for a train. Still not splitting it. Split! Split it! No! Dang it. Okay, I guess we'll just wait for another. Split this car. I just need to find something solid. Something really solid. We'll get this off. This wire is being awfully dang right. Such a weak flame.
Be fearful. I will set you on fire. Ah!